Okay, so again, I think you guys can all hear me now. I heard a couple of yeses, see a couple of yeses.
Yes.
All right, and you can see me?
Yes.
Yes.
Okay, sorry for that. Again, we're still in a learning phase here, trying to figure all this out. Luckily, my husband is way more tech savvy than I am, and so I might have to run down the hall and grab him a couple more times as we continue on today. But hopefully I will get this running smoothly here pretty quickly, okay? All right. Okay, so let's see, where should I even begin? So I think I have everyone on mute, and you have the ability to unmute yourself if you have a question. So what I think I'm going to try and do with these Zoom sessions is try and go sort of as slowly as possible, and then pause as often as possible for questions. Because again, given the class size, I'm not quite sure how easy it's going to be necessarily for you all to jump in at the same time and get your questions in. There is also, I believe, a raise hand function on in here. So you might be able to try that, and I can try and see if I can get your attention there too, or you can get my attention there too. Okay, let's see. All right. Well, what I would like to do actually is maybe just start with any questions of the material that I've posted already before we jump into today's lecture. So are there any questions about any of the things that I have posted already?
Yeah, I was wondering what would be covered in our next exam, which I see that it's in May. That's our next exam. Is that correct?
That is correct. So we will only have, yeah, our next exam. You know, I've got to go back and look at the syllabus because with my 3 classes, I can't remember which one I did what to. My recollection is that we will have one more exam, but I've got to double check that. So let me get back to you on that, and I'll send a message out with that information. How about that?
Okay, thank you so much.
Okay, any other questions? You can go ahead and unmute yourself if you have a question. No other questions? Okay, so then what we'll do is we'll go ahead and jump into the material then for today. If of course I can figure out how to get there. There we go. Okay, so can everybody see the slide Parasitic Diseases?
Yes.
Yes, okay.
Yes.
Great. All right, perfect. Okay, so what we began discussing, or what I began discussing in the last lecture, was trying to put all of the puzzle pieces together, where we've spent this time talking about the innate immune response, the adaptive immune response, and then lastly, the complement system. And again, it was to try and put all of the puzzle pieces back together again, where we look at the individual pathogen categories, talk about the immune response in general in response to those different pathogen categories, and then look at some of the evasion mechanisms that we know exist with certain pathogen categories, okay? All right, let me just make sure I've got everyone in. I've got a few late participants. Okay, I think I got them all in. All right, so in the last lecture, the one that was posted, we had talked a little bit about bacterial pathogens, and then we talked a little bit about viruses. And so with today's lecture, what I'm going to do is transition into parasitic pathogens, and then a little bit about fungal pathogens as well. So as you can see on this slide right here, we've got parasites are organisms that grow, feed, or are sheltered on or in a different organism, basically contributing nothing to the survival of its host. So when we talk about parasites, parasites fall into this particular category overall. Now, when we discuss parasites, there are two broad categories of parasites that we can focus on. There are those that are single-celled parasites, and as you can imagine, our immune system responds differently to those single-celled parasites in comparison to the second category, which are multicellular parasites. Multicellular parasites, as you can imagine, tend to be much larger than our single-celled parasites, and our immune response has a different response overall to these larger multicellular parasites. What you can see on this slide are just a few examples of single-celled parasites. Okay, so I think I did actually get a hand. So Jesse, did you have a question?
Where is this PowerPoint?
This PowerPoint should be in Blackboard. Did
I can't
else
find it.
You did not find it?
No.
Okay, so I will make sure after class that I go back and I add that PowerPoint back in. I thought I had done it, but I apologize if I did not do that. So nobody else was able to find it?
No,
No.
Okay, I apologize. As you can imagine, again, we're trying to juggle 3 classes and be a mom all at the same time has been a little bit chaotic for me. So I'm trying to get it all under control. Let me go ahead and I'll make sure that that PowerPoint is posted for you as soon as class is over today, okay?
Okay, and is this class also going to be recorded as well, or?
Yes, I am recording it right now so that as soon as I am done, I will get it uploaded. It'll be on YouTube so that you can then access it and go back and listen to it just like you can the other lectures that were recorded.
Awesome, thank you, Professor.
You're welcome. Okay, any other questions? No. Okay, so the raise hand function does seem to work and it gets my attention. So I will keep my eye on, this screen as well. If you have a question, go ahead and raise your hand and then give me a second to get to you. And I will hopefully be able to address that for you. All right, so back to this particular slide. What you can see here are a number of examples of single-celled parasites. These single-celled parasites do fall into the category known as protozoa or considered protozoans. And again, as you can see here, there are a number of examples. So we have something like Giardia, which actually causes gastrointestinal disorders. We have Cryptosporidium, which also causes gastrointestinal disorders. We have Toxoplasma, which causes a flu-like disorder. Entamoeba histolytica causes amoebic dysentery, also a gastrointestinal disorder. Trypanosoma cruzi, which is the last one on the figure overall, causes African sleeping sickness. Probably the #1 single-celled parasite that you may have heard about previously is a parasite known as Plasmodium, and it is the parasitic agent that results in the disease known as malaria. And it turns out that malaria is the #1 parasite-induced death, causes the let me take that back. Plasmodium causes malaria, and malaria is the #1 parasite-induced death worldwide. Okay, so questions about this slide before I move on? No, okay. All right, so like I just mentioned, malaria is caused by a single-celled organism known as Plasmodium. There are a number of species that do in fact cause malaria. Plasmodium is the genus for this. And what you see here is the life cycle of Plasmodium. Now, Plasmodium is transmitted by mosquitoes, and they basically mate and reproduce in the mosquito gut. And when the mosquito then lands on a human, because it's drawing up the blood of the human, actually transmit or passes these components known as the sporozoites. The sporozoites don't stay in the blood for too long. They actually migrate to the liver, where they live for a period of time in the liver, where they then change their life cycle stage into merozoites. The merozoites can actually leave the liver, and they do in fact infect red blood cells, so they get inside the red blood cell overall. The life cycle continues into the gametocytes, and then again you see down at the bottom of the figure the mosquito then taking again another blood meal, picking up those gametocytes. They end up in the gut, and again the cycle continues. What I want to say is the most compelling component about why parasites, single-celled parasites, for example, are actually as successful as they are, is because they undergo something known as antigenic variation. This particular slide is a really great example of that, in that there are different phenotypic physiological components associated with different stages of the life cycle of this particular parasite. So as you can imagine, if our immune systems are trying to detect antigens for one life cycle stage, then the life cycle stage changes. Antibodies, for example, that might be produced against that first life cycle stage no longer have that antigen present to bind to overall. And so this antigenic variation, this changing of life cycle stages, and in particular antigens that are present in life cycle stages, make parasites, single-celled parasites, actually very successful overall. Okay, so questions about this slide? No? No questions? Okay. So in this particular slide, this highlights a little bit more of that antigenic variation. This happens to be for an organism that causes African sleeping sickness. And what you see here, what I think is really nicely done with this slide, is to show you different antigens that are present in different stages of the life cycle of the organism, indicated by the blue color, the pink color, and the green color overall. So what happens is that during the life cycle changes, the antigens also are changing, so that again, antibodies that our immune system produced against the very first component are essentially not going to work, because by the time the antibodies are produced, the antigens have changed, the life cycle of the parasite has changed, and so you get a different antigen that's present that the first antibody doesn't bind to and recognize and interact with. And this is why, again, these organisms tend to be so successful. So questions about this slide? No? No questions? Okay. All right, now parasites can also be multicellular. So again, the two broad categories of parasites are the single-celled parasites and the multicellular parasites. Now, multicellular parasites can be things like, on the right hand of the slide, a louse, a tick, a flea, for example. And our immune systems do in fact have a minor response against them, but these are components that actually live on us and not getting into the host system. What we really want to talk about are those parasites that have an ability to get into the host system, just like any of the pathogens that we've discussed previously. And those most commonly are going to be parasitic worms. So parasitic worms fall into multiple categories. There are those that are referred to as roundworms, and these are things known as nematodes. We also have flatworms, which include things like flukes and tapeworms. So flukes are worms that can be found in tissues of the host, and tapeworms are typically multicellular parasitic worms that are found inside the gut. Okay, so questions about this slide, just sort of highlighting the difference between single-celled parasites versus multicellular parasites. No, no questions. Again, if you have a question, if you could use the raise hand function, that would really help me out quite a bit. Okay, everybody still with me?
Yes.
Okay, great, thank you. Again, a little odd to be doing it this way. You guys might have more experience with some of your other classes, but for me, I'm still trying to learn these ropes a bit. All right, so let's go ahead and transition then to the next slide. Okay, so this is where I was saying what we want to do is sort of put all of the pieces of the puzzle back together again. Obviously, we've spent the first half of the semester talking about the different immune responses, and so this is really just a little snippet overall, but these are some of the specifics that are occurring in response to these parasitic types of infections. So up at the very top of the slide, what you see is that there is an increase in IgE class antibody production. So the IgE class specifically is the class that does in fact play a role in warding off parasites. When we talked about antibodies and antibody classes, this is how we described IgE, and that its job is to play a role in fighting off parasitic infections. We also know that IgE, when there's problems, play a role in hypersensitivity reactions, but that'll be something we'll talk about a little bit later. So sort of the normal everyday role of IgE is to help fight off parasitic infections. We also see that there's an increase in mast cells at the target location, and it is IgE that binds specifically with mast cells to cause what's known as a degranulation event. So mast cells are granulocytes. They contain these granules that have different components. Most importantly, what we find in these granules are things like histamine, which hopefully you recall a little bit about histamine. Histamine's job is to play a role in allowing for the inflammatory response to take place. In addition, mast cells contain heparin in their granules, and heparin is an anticoagulant. So this just allows the blood to flow relatively smoothly so that we can have the migration of immune cells to the target location where an infection has taken place. Let's see, sort of in the middle of the slide, you can see obviously the adult worm. You can see the antibodies that are binding overall. But what I want to highlight is that there's also an increase in eosinophils. So again, way back at the beginning of the semester, when we talked about different cell types, we discussed the fact that eosinophils' sort of number one job is to play a role in fighting off parasitic infections. So it's no surprise that we're seeing them in a really big role at this particular point. So what the eosinophils do is, in conjunction with the antibodies, is elicit the antibody-dependent cell cytotoxicity mechanism, ADCC. We discussed ADCC a few times in the semester, in particular looking at natural killer cells and their job in initiating ADCC. But there are other immune cells that can participate, including eosinophils. So eosinophils have the ability to initiate the ADCC response. And as you can see in this figure right at the center, there's the eosinophil interacting with the antibody to then trigger that response. Now, the reason that multicellular parasites are actually a very big problem for our immune system is that these are multicellular organisms. And so being multicellular, they're going to be much larger than any of the cells that are part of the immune system or the immune response. And so phagocytosis really doesn't happen here because you cannot phagocytose something that is way bigger than you are. And so something like ADCC might help trigger some cellular damage sort of on the exterior surface of these parasites to hopefully cause the demise overall of the parasite. Okay. So questions about that so far? Okay, Richard.
Yeah, so what did you say happens when the IgE combines with the mast cells?
So when the IgE, up at the top, when IgE combines with the mast cells, it causes the degranulation of the mast cells. And the granules, again, contain a number of things, but most importantly, histamine and heparin get released. So the histamine helps with the inflammatory response. Heparin allows for, is basically an anticoagulant to allow the blood to flow still really smoothly at this point. So did that answer your question?
Yes, thank you.
Okay, you're welcome. All right, so what we then see with the inflammatory response is an influx of additional cells to the site of the infection overall. So you can see a number of cells coming in sort of from the bottom and where the blood vessel is. You've got macrophages coming in, mast cells recognizing some of these signals to then allow for additional cytokine production. And that additional cytokine production will again help proliferate the immune response overall. All right, so questions about that? All right, I'm actually going to jump back to the single-celled parasites just for a minute because I left out a whole bunch of stuff that I wanted to talk about. So we're gonna move back just a little bit here. Okay, so just to regroup myself here. The point here is that with parasites, there are the two large categories. There are the single-celled parasites and the multicellular parasites. So single-celled parasites do present a problem. They can be extracellular, but there are some parasites that can be intracellular as well. So there are a whole host of different types of responses for our immune system that need to come in and take place. So any of the things that we've discussed previously for extracellular components would definitely hold true, as well as the intracellular components. When we talk about intracellular parasites, all of the things that we've discussed when we have responses that target altered self cells, this would be a case of an altered self cell. So previously we've discussed altered self cells, and the two examples I always use in class are cells that are infected with the virus or cells that are cancerous, right? Those are the two main categories of altered self cells that we've discussed. Well, in this particular case, this is another example of an altered self cell. If the parasite is one of those types of parasites that can actually get inside of a cell, well, now we have a need for that type of response to then target those altered self cells for destruction. So I just wanted to clarify that really quickly. So questions about that? No, okay. All right, so in addition to that, I did mention the fact that these parasites tend to be very successful, even these single cellular ones, or in particular these single cellular ones, because they do undergo this mechanism of antigenic variation. They change their physiological structure, their antigens that are present throughout their life cycle, and because those antigens change, again, an antibody that might be produced against one antigen, that antigen may no longer be there as the parasite transitions into different stages of its life cycle overall. What I also wanted to do in coming back to this slide is just highlight a few mechanisms of evasion aside from antigenic variation. Now, antigenic variation is probably the #1 evasion mechanism for single-celled parasites, but there are some other examples. So for example, for Plasmodium, that causes malaria, there is an additional protein component that is on the extracellular surface that blocks antibody binding. So you can see that that would be a pretty strong evasion mechanism, if antibodies are unable to bind to the parasite, all of those antibody-mediated responses would then be negated. For trypanosoma, trypanosoma causes sleeping sickness. It too also undergoes that antigenic variation I was just talking about. But there are some other examples of evasion mechanisms. It turns out that this particular parasite can actually produce molecules that are very similar to cytokines. Now, you might go, well, how is that an evasion mechanism? Because cytokines are supposed to help the immune response. Well, it turns out that cytokines are signaling molecules that do help the immune response, but these cytokine-like molecules that trypanosoma can produce alter B cell and T cell development overall. And so by altering T cell and B cell development, you then sort of eliminate some of those downstream responses as well. One more evasion mechanism that I wanted to highlight was that for Entamoeba histolytica, which is the fourth one over on this particular slide. This particular organism actually produces a protease that has the ability to degrade antibodies. So again, what you can see here is a variety of different types of evasion mechanisms that allow these parasites to be successful. So questions about that? Okay, Ariel, you can unmute yourself and ask your question, please.
Yeah, my question was just, did we have to remember the parasite that goes with the evasion mechanism?
If you have, yeah, probably the genus and then the evasion mechanism would be good to know. In general, though, what I'm really interested in is that you have an understanding of what these evasion mechanisms are. So the specifics of which species and which evasion mechanism, probably not so important, but the grouping of single-celled parasites and their evasion mechanisms is probably more important.
Okay, great, thank you.
Okay, you're welcome. Okay, any other questions with this particular slide and what I just kind of went back to review? No? All right, so let's continue on. All right, so we kind of went through this slide and we discussed what happens in general with an immune response against a multicellular organism. So then the next thing I would like to do is talk about evasion mechanisms. And in this particular case, there's really not much to discuss. These are multicellular organisms. They, depending upon the species, can be rather large. If you think about a tapeworm and any kind of gross YouTube video you could come up with looking at these tapeworms, they are relatively large. Now again, there are some smaller worms that can infect host systems. But the point here is that these are multicellular organisms. In general, they're going to be much larger than any of the single-celled immune cells that are trying to ward off the infection. So they don't really need to evade the immune system in any way, simply because they're so large. That right there is a reason why they're so successful overall. Again, no phagocytosis is going to occur here because these are all multicellular organisms, and we know that phagocytosis is a really potent and important component of the immune response, and that just doesn't happen with these large multicellular types of parasites. So questions about this one? Ariel, did you still have a question?
No, sorry.
That's okay. I went to a meeting just the other day where we all left our hands up the whole time. So if you just, once you're done, you can then like lower your hand. There we go. Perfect.
Got it.
No problem. We're all learning. Okay. So any other questions with the multicellular parasites? No questions with the multicellular parasites? Okay. Oh, and I just realized I added all kinds of animation that I was going to use as I went through. So now it's too late, but that's okay. There's the animation. All right. So let's transition into the last category of pathogen, and those are fungi. So fungi and fungal diseases represents a very diverse group of organisms. The diseases that are caused by fungal parasites are collectively known as mycoses, like you see on the slide here. And this particular slide just highlights a number of different locations where fungi can actually enter into the host system and cause problems overall. It comes from a review article that was published in 2012. And I'm never going to ask you all of these details. I really just want to make sure that you're aware that there are, in fact, a number of different fungal pathogens and disorders that are associated with fungal pathogens. What I do want to discuss just a little bit is how are they classified? So because there are so many of them, like we've talked about previously in the semester, is when there's a lot of information, scientists like to categorize it and put it into neat compartments. So one way to classify mycoses is by the site of infection. So where exactly does the infection take place? So that could be something that's superficial, for example, on the epidermis. It could be cutaneous, in the skin or the hair or the nails. It could be subcutaneous, entering into a wound. Can actually be deeper systemic, like the lungs and abdominal tissue. So I think everyone is probably familiar with valley fever. Valley fever does, in fact, cause the disorder where the fungal pathogen coccidioides enters into the lungs. And so that would be one of those deeper systemic kind of categories for site of infection. Another way to categorize them would be the route of acquisition. So how did they actually enter into the host? Were they exogenous, something like environmental overall, airborne, cutaneous, or are they endogenous? There are some fungal components that are commensal, meaning that they live in us and don't really cause us any harm under normal conditions. However, when our microbiota kind of goes out of whack, that sometimes allows for these commensal fungi to then take over and cause problems overall. Something like a vaginal yeast infection, for example, is a good example of that. This is a type of fungus that is commensal and doesn't typically cause problems except for when our microbiota goes out of balance and then can sort of take over and cause problems. The last way that mycoses can typically be characterized or classified is by their virulence, and that is they could be considered primary, which is extremely virulent, infects healthy hosts, basically infects just about anybody, or it can be opportunistic. It has very low virulence and usually just infects an immunocompromised host. So again, the point of this particular slide is just to highlight the fact that there are lots of different fungal pathogens with lots of different diseases that are associated with them, and then just talk a little bit about ways that we can classify them. So questions about this slide? No questions about this slide? Okay. All right, so what makes fungal pathogens, I guess, so successful overall? It turns out that fungi, very similar to what we just talked about with the single cell parasites, undergo variations in their exterior phenotype or their exterior appearance during different stages of their life cycle. Now, because of that, we can also have that phenomenon known as antigenic variation, because antigens that might be present under one life cycle stage may not be present in a different life cycle stage. Again, our immune systems are playing catch up all the time. Our immune systems recognize, build antibodies against the first category of antigen, yet by the time those antibodies are produced and potentially could do some good in fighting off the fungal pathogen, the life cycle stage has changed and different antigens are then present. So the antigen that might have been present initially is no longer present, so the antibody basically is not effective. So there are plenty of examples of these fungal pathogens that undergo these changes in antigen presentation because of changes in their life cycle components overall. All right, so questions about that? So again, antigenic variation, very important for fungal evasion overall. No questions? Okay. Oops, let's go back. Okay, so what I want to talk just a little bit about, there's unfortunately not a really nice summary slide like we had for the parasitic pathogens overall, but we do know that there are lots of different components of the immune system or the immune response that kick into play when a fungal pathogen is detected. For fungal pathogens that are still single-cell fungal pathogens or relatively small, phagocytosis is in fact a really important part of the immune response. So phagocytosis, again, we've talked about over and over again, and how important it is in the immune response. So there are in fact PRRs, pattern recognition receptors, that interact and bind to certain fungal cell wall components, or can in fact recognize and interact cell wall components. And it turns out that these PRRs do in fact do a really good job at controlling fungal infections, with phagocytosis again being the primary response. When we discuss the complement system, we also discussed the fact that in the complement system, there were 3 different activation mechanisms. Both the alternative and the mannose-binding lectin activated pathway do in fact recognize cell wall components of fungal pathogens. So the complement system too is playing a role in detecting these components and playing a role in fighting off these fungal pathogens. We do know that there are examples of antibodies that are present, that are built up against different types of fungal pathogens. So the adaptive immune response is also playing a really big part in fighting off these infections. Okay, so then let's switch gears just a little bit and talk about the evasion mechanisms. I highlighted that already for you, and that probably the #1 evasion mechanism is this antigenic variation. Different stages in life cycle have different antigens that are present, and so our immune systems are always trying to catch up in being able to detect the pathogen overall. There are some fungi that have the ability to adhere to the host cell surfaces or tissues really strongly, and it is this fungal adherence that plays a role in making them very successful as well, because they sort of stick to the tissues, and by sticking to the tissues overall, that then plays a role again in helping them evade the immune response. Some fungal pathogens also produce a capsule, which is an extra polysaccharide coating that is on the exterior surface of the cell wall. And by having this capsule, this polysaccharide coating on the exterior surface, that then makes them sort of slick and slippery, and also makes it very hard for PRRs to bind. And if PRRs can't bind, then that whole phagocytic mechanism that we were just talking about is sort of bypassed as well. In addition, there are a number of fungal pathogens that produce proteases. Proteases, again, large families of enzymes that degrade proteins. In particular, they can play a role in degrading antibodies. And so those are some of the fungal evasion mechanisms overall. Questions about fungal immune response and then fungal evasion mechanisms? Maria?
For the capsule part, it was a polysaccharide coating, you said?
Yes.
Okay, and it how does it work again?
It basically is on the exterior surface of the fungal pathogen, and it prevents PRRs from binding to the fungal pathogen.
Okay, thank you.
You're welcome. Okay, any other questions about that? David?
So would you say that the innate, the adaptive, and the complement system all work against fungal pathogens?
Yes. Yes.
Okay, and in parasites, innate immunity doesn't do anything, or how does that work?
No, it does, but it depends on the class of parasite. Again, when we're talking about a single-celled parasite, the innate immune response does in fact have a way of kicking in, and that phagocytosis mechanism does kick in. But when we're talking about a multicellular parasite, then there really is no phagocytosis that's going to take place.
Okay, that makes sense. Thank you.
You're welcome. Okay, other questions? All right. Okay, so lastly, what I want to talk about just really briefly is the fact that we have emerging infectious diseases, and this is kind of timely with what we're going through right now. I hadn't thought about it from that point of view until just looking at the slide again today. But the point of this particular slide is to really talk about the fact that we have what's highlighted in the black are re-emerging diseases. So these are things that once were under control and seem to be sort of making a reappearance worldwide. Now this oftentimes is due to globalization, interactions with people and wildlife, anti-vax movements, that type of thing. And so we are seeing sort of a reemergence of certain diseases that we kind of thought were under control. And then in the red are the newly emerging diseases. These are all things that are new to us and cause or have caused severe problems worldwide. And if you look at this particular slide, sort of on the top right corner is SARS. And so that fits in really nicely with what we are currently dealing with, in that newly emerging diseases are definitely a problem overall. And why they are such a big problem is that if our immune system or our immune responses have not interacted with these pathogens previously, we don't have something that is referred to as herd immunity. And so we'll come back to this concept with our next set of information dealing with vaccines. But herd immunity is basically a phenomenon where people are protected when approximately 70% of the population is immune. Now that can be due to vaccines, for example, but it can also be due to previous exposure. And because we have something, again, with the coronavirus that is something brand new to all of our immune systems, our immune systems are having a really hard time interacting with them. And we don't have sort of an a herd immunity type of situations where individuals have been exposed and survived. Now, hopefully we're going to get to that point where we do have individuals who survive and have an immune response built up, particularly those memory cells that can then fight off an infection when a repeat exposure comes into play. But at this particular point, we are dealing with that phenomenon right now. Okay, so questions about this particular slide? Yeah, Sam.
In the bottom right-hand corner, where it says just plague in general, pointing to the middle of Africa,
Yeah.
What does it mean by plague?
Well, plagues are general terms that are utilized. Usually a bacterial pathogen is associated with a plague. I'm not sure what that's referring to necessarily, because that could be something that's worldwide, not necessarily just Central Africa.
Okay.
Okay, other questions? Okay, so what I wanted to take a few minutes to do, maybe just a minute or two, is again, open it up to if there are any other questions that you might have on the previous material. I know I have a sort of a notes here for myself just to make sure that as soon as we're done, get this PowerPoint posted for you in Blackboard, and I will do that immediately. There was also a question about the material that will be on the next exam, and so I will go back and review that from the syllabus and then sort of send out a summary update on that as well. Are there any other general questions that you might have?
Yes, I have a question.
Yes, go ahead.
I know you're recording this lecture. Are you going to upload it right after the class is over?
Yes, so I will get the PowerPoint loaded, and then I will also, it takes a little time for it to download and then to process, but as soon as that's done, I will upload it for you.
Okay, thank you.
You're welcome. Okay, any other questions? No? No other questions?
I do have a question about your office hours.
Okay, go ahead.
I know, are you still having the same time as your office hours? And if you are, do you recommend us still using this Zoom link, or do you have your own?
What you will need to do is just send me a message that you would like to meet during Zoom. So I had originally thought that I would just kind of sit here and wait for people to show up, but I decided against that. So I'm available if you have questions, definitely during my office hours, but just let me know and I'll definitely be here for that. But if for some reason you can't make it during those office hours and you'd still like to have a Zoom session, we can set up a separate session for sure.
Okay, thank you so much.
You're welcome. And also when questions come through, I think, yes, for this class, I've already had a couple questions where I compiled the questions and then I repost. No, maybe it wasn't this class. Was it genetics? Might have been genetics. Sorry. Again, classes are getting mumbled up in my head a little bit. But as questions come in from you guys, I will compile them and then I'll post them so that you can have access to those as well. You can also use the discussion board to ask a question, because sometimes, you know, you might have a question and someone else might have that same question, and it's just good to get those kind of things answered. So you can contact me, we can set up a Zoom session, you can just email me with the question, or you can put it in the discussion board, and I will try to attempt to check all of those things to make sure I get your questions answered. All right, any other questions? No, I hope you guys are all staying safe. I know this is a really, really crazy time. It's a crazy time for all of us. But in particular, if you haven't read the latest update, the next 2 weeks are probably going to be the worst of what's going on. And if you don't need to be out, don't go out. There's even recommendations to stay away from grocery shopping. If you don't need to go grocery shopping, have it delivered if you can. Try and stay as safe as possible, okay? And I'm thinking about all you guys. I hope to see you guys again sometime soon. If there's no other questions, then we'll go ahead and end this. And like I said, I will get the PowerPoint posted. I will get this recording posted, and we'll go from there.
Thank you. Stay safe.
Thank you.
Thank you.
Bye, guys.
Thank you. Be safe.
Take care.